Now let's examine what is the activation signals that generate acid in the stomach. In other words, what signals tell the parietal cell to begin HCl production? Well, these are multiple levels of information that the parietal cell receives. The first one to consider is the structural architecture of the cell. If this is a parietal cell, of course, the apical surface is going to be facing the lumen of the stomach. Consider the fact that acid production has to occur rapidly after food is ingested. There's two ways to generate the protein to function in this process, this protein being the proton potassium ATPase transporter. We can do de novo synthesis, which means that you could potentially activate a gene to make mRNA, translate that mRNA into protein, and then traffic that protein to the luminal surface. Now consider how much time that takes. This process of gene to protein, to have a sufficient amount of protein expressed so that there's a functional difference, can take approximately 14 hours. Now in our previous graph we saw that the acid production began in minutes after food consumption began. So that argues for a predisposition of protein in vesicles. So if you look at a parietal cell under a microscope, you will see a very, very large number of vesicles that appear to be positioned largely underneath the luminal surface of the cell. These vesicles all contain the potassium, proton, ATPase transporter. So this means that this protein has been pre-made and pre-packaged in these vesicles, so that upon stimulation of the parietal cells, these vesicles now move to the plasma membrane on the luminal surface, fuse, and now expose this transporter on the surface so it can function and transport protons. So let's look at some of these signals that begin this process. If we consider a large section of tissue in the stomach, let's say that here is our parietal cell, and this parietal cell is generating our acid. What stimulates this process? Because we don't want this process to occur when there's no food present, or there's no sense of food becoming present. Otherwise, we will waste energy producing acid and continue and possibly cause damage to the stomach lining. So we need a controlled process. Well, the first process is going to be vagus nerve stimulation. Now recall back in the salivary gland, vagal stimulation occurs at the beginning when food is sensed. So when you either visualize it or see it, we get the signal from the vagus nerve to begin the digestive process. This is part of the cephalic phase of digestion. So the vagus nerve is going to stimulate through acetylcholine transmitter the parietal cell to begin the process of acid production. Now, as you can imagine, this is not an overwhelming signal because there's no food present in the lumen of the stomach yet. So this is just preparing the stomach for the possibility of food because you have seen it or you have smelled it. This is the Pavlov experiment. Now the next level is an endocrine method. So we have to introduce the concept of enteroendocrine cells. These are endocrine cells located in the body, and they are responding to stimuli and producing hormones. So the first one that we have to consider is what's called the G cell. The G cell is responsible for sensing amino acids. And peptides. So these, of course, are coming from dietary source, being ingested. We have some preliminary digestion of the proteins we consume. These trigger cell surface receptors that now stimulate the G cell, and the G cell is going to secrete the hormone gastrin. Gastrin will now enter the blood and gastrin will stimulate directly the parietal cell. Gastrin stimulation of the parietal cell is a significant increase in acid production. Once we have this, now we enter what's called the gastric phase of digestion. So we've transcended from the cephalic phase to the gastric phase. Now the gastrin works through CCKB receptor. We'll talk more about CCK later on. This is cholecystokinin. This is also another factor, another hormone that's produced in the digestive process. We will see this in a bit later. But this receptor, the gastrin receptor, was named CCKB because it's related to the CCK receptor. Now, the other process here are mechanoreceptors that are present on the luminal surface of the stomach, and these will stimulate G cell activity. So how do we stimulate a mechanoreceptor? We eat a Thanksgiving meal. We stretch that stomach out. That activates. That is a secondary signal to the G cell in addition to the dietary components. Finally, the other cell type we have here is called an ECL cell, for enterochromaffin-like cell. This cell is also an enteroendocrine cell, but this one is considered closed, whereas this one is considered open. Open simply means that it's open to receiving dietary signals, being open to the body in the lumen. Closed means it's completely embedded in the tissue and has no contact with any sensing of dietary components, but what it is sensing is gastrin. So gastrin is a stimulant of ECL cells, as well as vagus nerve stimulation, and the ECL cell will produce histamine. And now histamine is going to act in a similar manner as gastrin to stimulate the parietal cell, and it does so through the H2 histamine receptor.